Imagine putting a person madly in love into an fMRI scanner to discover their brain activity perfectly mirrors a cocaine addict. Then, in a separate lab, researchers pinpoint a single hormone receptor that dictates whether a small rodent will mate for life. Meanwhile, a mundane laboratory questionnaire designed by psychologists inadvertently creates multiple real-world marriages between total strangers. Peeling back the layers of these biological and chemical mechanics can feel a bit cold, but understanding the exact neural structures governing your emotions doesn't make your personal experience of them any less real or meaningful. It simply relocates the mystery. Biological anthropologist Helen Fisher defined romantic love as three distinct, evolutionarily separate brain systems operating in tandem. Looking at this model, we can trace the three separate tracks. The first pillar is lust. Driven primarily by testosterone and estrogen, its evolutionary purpose is to broadly seek out mating opportunities. The second system is attraction. This is the intense, passionate early stage of romance, fueled by dopamine and norepinephrine, designed to focus all your mating effort onto one specific person. The final system is attachment. Driven heavily by oxytocin and vasopressin, it creates a calmer, longer-term bond oriented towards sustaining a partnership long enough to raise offspring. Because these three systems possess entirely separate neurochemistry, they can operate independently of one another. You can feel deep attachment to a long-term partner, intense attraction to someone new, and lust for entirely different people, all simultaneously. Human romance feels chaotic because your brain is running three different programs at once. In 2005, Fisher and her colleagues decided to map that second system, attraction, in real time. They placed 17 individuals who described themselves as intensely in love for roughly 7 months into MRI scanners, and showed them alternating photographs of their partners and neutral acquaintances. This simplified cross-section of the human midbrain shows exactly how the brain reacted. Viewing a partner's photo specifically activated two structures, the right ventral tegmental area and the right caudate nucleus. These regions are the richest sources of dopamine-producing neurons in the entire brain. This is the reward circuitry activated by cocaine and other addictive substances. But the high of dopamine is only half of the equation. There is also the subjective experience that nearly everyone who falls in love recognizes, the complete inability to stop thinking about that person. A separate study, led by psychiatrist Donatella Morazzetti, analyzed the blood of people who had recently fallen in love. The data showed that their levels of the serotonin transporter had plummeted compared to psychologically healthy control subjects. When Morazzetti mapped these severely reduced serotonin levels, she found they were statistically indistinguishable from those measured in patients diagnosed with obsessive-compulsive disorder. The biochemical evidence points to a stark conclusion. Early-stage romantic attraction functions neurologically as a genuine behavioral addiction, complete with intense craving, paired directly with a clinical obsession. To understand how attraction transitions into long-term attachment, science turns to the prairie vole. It is part of the small minority of mammal species, under 5%, that forms lasting monogamous pair bonds. The prairie vole shares its habitat with a nearly identical cousin species, the meadow vole, which is entirely promiscuous. Yet both species produce the exact same attachment hormones, oxytocin and vasopressin. This side-by-side comparison reveals the physical mechanism responsible for the behavior. Monogamous prairie voles possess a vastly higher density of hormone receptors packed into their reward circuitry, allowing their brains to absorb the incoming molecules much more efficiently than their promiscuous cousins. Researchers proved this by artificially increasing the receptor density in the promiscuous vole species. Doing so successfully induced long-term pair bonding behavior in animals that do not naturally bond at all. Around the time biologists were untangling rodent genetics, two graduate students named Arthur Aron and Elaine Spaulding met at UC Berkeley in 1967. The couple eventually married and established an interpersonal relationships lab to study intimacy. In 1997, Aron and his colleagues published a study where they paired up strangers in a laboratory. The participants took turns asking each other 36 escalating, increasingly personal questions over 45 minutes, followed by 4 minutes of sustained, silent eye contact. The specific escalating structure of self-disclosure successfully generated deep interpersonal closeness. But Aron built the questionnaire solely to solve a logistical problem: how to reliably force two strangers to feel close to each other quickly enough to study them in a controlled environment. The methodology remained an obscure academic citation for nearly 2 decades, until a 2015 New York Times column published the questions for a mass audience. The experiment rapidly went viral, turning a sterile lab methodology into an informal cultural tool that has since produced multiple real marriages. Whether it relies on a dense cluster of physical hormone receptors in a rodent's brain, or a highly structured pattern of vulnerable self-disclosure between two humans in a lab, long-term bonding is built on specific, manipulable mechanisms. Viewing love through the lens of addiction forces us to confront the inevitable dark side of the framework. What happens to the brain when the object of that intense craving is removed? Helen Fisher conducted a subsequent series of MRI studies, this time placing recently rejected lovers into the scanner. She then showed them photographs of the very person who had broken their heart. The resulting brain scans lit up in regions associated directly with both literal physical pain and acute drug withdrawal. When you experience romantic rejection, your nervous system processes the loss as a physical injury. Heartbreak is a genuinely measurable neurological reality. A rodent's hormone receptor density, an escalating questionnaire between strangers, and dopamine rushing through your midbrain. Science can locate exactly which brain systems and behavioral structures make love possible, but uncovering those mechanics doesn't make the feeling any less real or meaningful. Thanks for watching.